So Mustafa, Darby, and I are really happy to be here today to, I guess, remind the Ameriflux community of a fantastic open source resource that's out there for advancing our interpretation of eddy flux data across seasons and years. And that resource is PhenoCam, a network of power-mounted digital cameras for monitoring the phenology of vegetation across diverse ecosystems of North America and the greater globe. Our talk today is meant to be more or less a basic introduction to why phenology matters for FluxNet scientists, how cameras can be used to keep our eye on the state of the vegetation from one day to the next, and really what the PhenoCam network does and how it can help our science. More seasoned users of PhenoCam data might be more interested in our next week's webinar, which focuses more so on the open source tools that are available to access or wrangle PhenoCam data. But we hope that you stick around for today anyway, and hopefully get something out of our presentation. I haven't timed myself or anything, but I think I'm going to spend about 30 minutes with a introduction to PhenoCam, and that includes a brief Q&A session. Then what's going to happen is I'm going to hand things over to Mustafa, who's going to give a tutorial on navigating the PhenoCam website. And that will be followed by a second Q&A session. And so I think altogether today we'll be here for probably no more than an hour. And so we won't take away a whole hour and a half of your afternoons today. So what is phenology, and why is it something that we should monitor? Well, if I were to begin with a little anecdote, so when I myself was first learning about phenology, I would often get phenology confused with the term phrenology, which is basically the pseudoscientific study of bumps on the human head. But phenology, even if it sounds similar, is actually quite different than phrenology. Phenology is the scientific study of periodic biological phenomena in relation to climatic conditions, or referred to sometimes more colloquially as the seasonal rhythms of plants and animals. Examples of phenology are really diverse and broad, from the emergence of plants in the springtime and their flowering, to things such as insect metamorphoses, animal migration, the ripening of fruits and crops, and autumn coloration and senescence. Phenology is something that's all around us all the time. The general public is usually interested in phenology because humans tend to be fascinated by how the seasons change and how those changes manifest. And ecologists are interested in phenology inherently because it represents this point of intersection between living organisms and their growth environment. Today, there is perhaps no better reason for us to study phenology than to understand the biological impacts of climate change. For example, there's now substantial evidence for spring phenological events, such as leaf out and flowering, occurring earlier in the year. And there are also a number of studies documenting autumn phenology events occurring later. These phenological shifts have been summarized by the IPCC reports, which have even concluded that phenology is perhaps the simplest process in which to track changes in the ecology of species in response to climate change. And all of this is really relevant to the science that we do in the Ameriflux and FluxNet communities, because phenological events such as the growth of new foliage in the springtime, it can have a significant impact on the physical properties of the land surface, from its albedo and aerodynamic roughness to its conductance to trace gases such as water vapor and carbon dioxide. Phenology thus plays a strong role in shaping land atmosphere fluxes, of mass, energy, and momentum, the growth and structure of the planetary boundary layer, and the broader general circulation and composition of the atmosphere. And the latter point there is actually really nicely illustrated by one of our favorite graphs, the Keeling curve. At the annual time scale, atmospheric CO2 concentrations at Mauna Loa in Hawaii declined from a peak in the springtime. And this occurs as the vegetation across the northern hemisphere makes that phenological transition from the wintertime to the springtime. So in summary, the rhythm of the seasons is really largely interconnected with the breathing of the biosphere that we always think about. How do we monitor phenology? Traditionally, phenology is something that has always been recorded by a human observer going out in the field and seeing how plants develop from one day to the next. And this is actually not much different than when you go out into your garden every morning to check on your tomato plant and see if that tomato is ripe enough to harvest for your breakfast. This traditional method to monitoring phenology, however, comes with the caveats that it's just not practical for applying over large areas, and it can also be potentially subjective. These limitations can be overcome with satellite remote sensing technology, which enables this global scale monitoring of phenology now based on seasonal changes in the spectral properties of the land surface and the land surface vegetation. However, satellite remote sensing comes with its own limitations too, such as interference by clouds and the difficulty to interpret the seasonal signal that we get from relatively coarse and often mixed satellite pixels. So my favorite approach to monitoring phenology is sort of a middle ground or an intermediate between these two approaches. And in my view, it really gives you the best of both of these methods, the best of both worlds. And that approach is what has become widely known as the PhenoCam method. This method uses digital cameras or PhenoCams to record time-lapse pictures of the canopy level vegetation. These pictures are captured at a frequency that enables a continuous monitoring of seasonal vegetation changes. And these pictures cover a scale that's intermediate between what you see on the ground and what is seen by satellite pixels. And that whole concept is depicted in this illustration on the right side of my slide here, drawn by Victor Leszek at Northern Arizona University, where the camera, which is located next to the eddy covariance system on the tower, lies between the observer on the ground and the satellite orbiting our Earth from outer space. So to provide an example of this, I have some PhenoCam pictures from the Bartlett Experimental Forest, a Miraflux site in the White Mountains of New Hampshire. From these pictures, we can watch the snow melt, the trees flower, their buds burst, and their leaves expand throughout the springtime. And then as the summer passes and we approach the autumn, we can watch the leaves change color and eventually drop to the ground. I'm going to play through these pictures one more time, and the second time I want you to pay attention to sort of the level of detail that you can obtain from each of the images. Notice that the extent of these images, or the field of view of the camera, is wide enough that we can get a sense of the canopy level, phenology, sort of, which covers an area that is similar to what an eddy covariance system might measure. But also notice that each image provides a lot of detail, enough detail for us to look at the changes in different species or even individual organisms. So each of these pictures that is captured by the camera and subsequently archived allows us to sort of go back in time and see what a site looked like at a specific place in time. And as we just saw, in a lot of cases, it's possible to determine the timing of phenological events just by visually inspecting the images. But it's actually also possible to extract quantitative information from PhenoCam pictures. And the secret to this lies within what's known as the red, green, and blue additive color model, which says that you can reproduce any color as perceived by the human eye through some combination of the three primary colors, red, green, and blue. This is actually exactly how old color television sets worked, which were made up of a grid of small red, green, and blue dots on the screen. And by varying the intensity or brightness of these dots, these color televisions were able to reproduce a wide range of colors. So digital cameras today, almost without exception, use the red, green, and blue color model 2 to produce realistic looking pictures. And this means that we can split PhenoCam pictures into these three components, red, green, and blue, for a particular region of the image. And then we can quantify the intensity of each of these primary colors. These values of intensity are represented by what are known as red, green, and blue digital numbers, which make up a RGB triplet. So folks out there who work with satellite imagery, this is essentially the same kind of procedure, where in this case the picture you take with your camera is like a 3-band digital image. The green chromatic coordinate is a color-based index that is widely used for phenological assessment from PhenoCam pictures, and it's basically the relative greenness of the image calculated from the RGB triplet. So on the bottom here, I have this graph shows the change in the green chromatic coordinate, or the canopy greenness, over 6 years, 6 full annual cycles at the Harvard Forest in Massachusetts. And what we can see here is a pattern that's pretty typical of deciduous broadleaf forests in the temperate regions. During the winter time, the trees don't have any leaves, and so the picture isn't that green. And then when their leaves emerge in the springtime, that greenness ramps up. As the leaves mature throughout the summer, their color turns from like this bright green to a darker green, and so the canopy greenness gradually declines. And then as the leaves change color and eventually drop to the ground in the autumn, the greenness abruptly falls back to that wintertime value. For certain applications, it might be of interest to know what parts of the annual cycle correspond the most closely to the occurrence of specific phenological events, such as leaf emergence in spring or autumn, color in fall. And to investigate, what's commonly done is a calculation of what's known as a transition date. These transition dates are calculated first by taking the range or amplitude of the greenness over the entire annual cycle, and then calculating the dates at which greenness reaches 10%, 25%, or 50% of that amplitude, both in the springtime and autumn. And so in these deciduous broadleaf forests, with validation against ground observations, we found that the 10% date corresponds the most closely to about when leaves are emerging in the springtime and when they reach their maximum color in the autumn time. So far, our focus has just been on red, green, and blue because these are the wavelengths of light that cameras see so they can produce realistic pictures. But a lot of digital cameras these days are actually sensitive to infrared wavelengths beyond the visible spectrum too. although these wavelengths are usually blocked or filtered out by the camera, so it can produce realistic looking pictures. However, some PhenoCams can leverage this capability to capture information in a fourth spectral band and calculate this pseudo NDVI index that is sometimes called the camera NDVI. And so the standard camera that the PhenoCam network uses, which I'll talk about in a few slides, has this capability to take sequential RGB color images and a monochromatic image that is sensitive to the infrared light. So with a single PhenoCam, it's possible to investigate how phenology changes through time at a single site. For example, across different years, or even from one decade to the next. But with networks of cameras, it would now be possible to investigate how phenology varies across broad gradients in climate and vegetation at the level of entire continents, as we know from networks such as Ameriflux. So in the last dozen years or so, a number of digital camera networks have been established for this exact purpose, including the PhenoCam network, which has operated with an emphasis on monitoring phenology across North America. The motivation for the PhenoCam network can perhaps be traced back to a FluxNet synthesis paper, in fact, from 2005, which encouraged FluxNet site PIs to install video cameras to record the state of the canopy each day. Later that year, on October the 4th, just before noon, an Axis 211 network camera took its first photo at the Bartlett Experimental Forest. And this site would later become the first site in the PhenoCam network. And on my slide here, you can even see that original PhenoCam, which is now archived on the top of the fridge in the Richardson lab at Northern Arizona University. By 2009, there are about 12 PhenoCam sites in the northeastern USA and adjacent Canada. And this included a handful of Ameriflux sites, such as the Harvard Forest, Howland Forest, and the University of Michigan Biological Station. And since then, PhenoCam has expanded into an entire continental level of phenological observatory, with now over 700 sites across diverse ecosystems of North America and the greater globe. These sites aren't just restricted to the deciduous broadleaf forests that characterize the northeastern USA, but we now have cameras in a number of evergreen needleleaf forests, drylands, grasslands, agricultural systems, and there are even cameras in ecosystems recovering from disturbance or on school campuses. And of these over 700 cameras, I checked this morning, and there are 412 cameras registered with PhenoCam with co-located flux data for investigating how phenology shapes or regulates land atmosphere fluxes. And Mustafa is going to show you how you can find those, those exact sites with co-located pictures and flux data later in his tutorial. But a huge thanks to the Ameriflux management project for updating the Ameriflux website earlier this year to have a tab showing which Ameriflux sites have PhenoCams, have co-located PhenoCams. One thing that I always emphasize in these talks is that Mustafa and I aren't the ones taking all of these pictures. But rather you, the community, are the ones contributing. And in this sense, PhenoCam has been strongly influenced by networks like FluxNet and being this bottom-up, cooperative network. The PhenoCam network's philosophy has always been, if you contribute imagery from your camera, then we're willing to archive it, process it, and make those pictures and data products available for the global community for free. We just require our collaborators to follow a standard protocol, and this includes using a standard camera with a standard configuration. So the standard camera that PhenoCam uses are the Stardot Netcam SC and Live 2 cameras, which are commercially available outdoor webcams that go for about $600 US. And to configure the camera, we've also developed a easy to use software tool called the PhenoCam installation tool, or the PIT. All imagery and data products are publicly available from PhenoCam, either as real-time provisional data or as an archive data release. So that's like a published data set with the DOI. And these data can be accessed from the PhenoCam website or using a number of open source software tools, which is going to be the focus of the tutorial section of today's webinar, as well as next week's webinar, which Darby is going to be leading. So to summarize and conclude, the PhenoCam network uses images from digital cameras, or PhenoCams, to continuously monitor the phenology of vegetation. These images provide a crucial link between what is happening on the ground and what is seen by satellites. And these data are suited for a number of applications, including studies of how the terrestrial biosphere is being impacted by climate change. And if I could add one final point to this conclusion, these data, we believe, are very valuable for the Ameriflux and FluxNet communities to use. These data can help us interpret eddy fluxes over timescales of seasons to years. They can help us evaluate remote sensing products, which allow us to scale fluxes across space. And the PhenoCam data can also help us improve terrestrial biosphere models, which allow us to look into the future. If you would like to contact us, I've thrown our contact information up here. And I guess I don't remember, Kung, if you mentioned, but Mustafa has set up a share folder that you can, I guess, access through this QR code. And I think Mustafa is going to paste the link in the chat.
Yeah, I just did that. Yeah.
Okay, thanks so much, Mustafa. So all of the materials from today's webinar, as well as next week's webinar, will be available in that publicly available folder. And it looks like that's my last slide. So what Mustafa and I are planning to do is maybe answer any questions that the audience might have now. And then we're going to move on to Mustafa's tutorial. Then we'll have a second Q&A question, second Q&A session at the very end. How does that sound? Okay, so I see. Thanks, Kung. I thought I saw a question in the chat, but actually it was just Mustafa sharing the link to the slides. So
I see one question on the chat.
Have you seen useful signal in diurnal change from the PhenoCams? Yeah, that is a question that we sometimes get. I see the canopy greenness changing over the day. Does that provide any meaningful information? And for the most part, we haven't found anything useful. Usually it's due to changes in the sun angle in relation to the camera and the canopy throughout the day. Mounting height. So I think I had a, traditionally the PhenoCams have always been mounted on the, on towers, tripods, the roofs of field stations to get a landscape level view of the camera. But that being said, the cameras can be mounted on different kinds of structures for different purposes. For example, on this slide here, we have a camera mounted a meter off the ground on a T-post, and This was actually a camera that was tracking a rainfall manipulation experiment. There are even PhenoCams mounted, near the ground so that they're pointing right at the buds of, branches. And so the answer is, really the mounting style of mounting can be adjusted for different applications. Usually, as long as the camera is not pointed sort of towards the sky, we found that the quality of data is usually pretty good.
So Oscar, there's another question about are there any other costs besides the $600 camera that would be essential to start a new PhenoCam site?
Thanks, Mustafa, for reading out that comment for me. So at the moment, PhenoCam has a strong preference for using our standard protocol, and that's because we know that it works. And while it's not, completely inexpensive, it is relatively inexpensive. That being said, in the past, the PhenoCam team has tried to address the question: does camera choice matter? And thankfully, our conclusion is that camera choice might be of secondary importance. And so, a PhenoCam could cost as much as $3,000, but could also cost as low as $100. For example, there's a number of groups, I believe, using, Raspberry Pi cameras and Pi cams, and those have been pretty successful as long as it's not for like long-term campaigns. And I've also seen some good data, come out of sort of game cameras or nature cameras, such as, Wingscapes is a sort of commonly used bird watching camera. And so
So I feel like Jennifer's question is also about like secondary costs. Like, is there any cost like for PhenoCam network or whatever, any other cost other than camera? So I feel like there is no cost for in on our end. So and it's totally free once you have your camera and you have a Wi-Fi or whatever at your site that can connect to the PhenoCam network at NAU. It's totally free on our end, and we are gonna process all of those images.
thanks, Mustafa. Yeah, perhaps I misinterpreted Jennifer's comments. So thanks for completing my answer. I hope that
Yeah.
answers your comment.
So there's another question, Oscar. Is the red chromatic coordinate RCC useful for identifying change in leaf color, or is just the drop-off in GCC enough?
I don't have any first-hand experience using the red chromatic coordinate, but um, in the but it has been used for determining the date of peak leaf color in those eastern deciduous forests or like a aspen forest or something. And I believe it has had equal or even better success in capturing those dates versus the drop off in the GCC. Mustafa, do you know anything about the RCC?
So I know there are some specific purposes for that, like for some kind of microbial activities and other things that people use RCC, but I guess in terms of phenology, I guess GCC, I guess is more popular at least. Yeah, I'm not totally sure.
The other thing is that the camera only has three bands, or it can only look at three wavelengths. And so there's a bunch of color transformations you can try, but at the end of the day, they're kind of similar in the end. Mustafa, any other comments in the
Yeah, there's
chat?
another question. Can RGB PhenoCam be used to estimate plant water use?
Yeah, I mean, I suppose on a seasonal to annual time scale, um, plants are generally, or ecosystems are generally more efficient at moving water returning water from the soil to the atmosphere when they have greater, more leaf area. And so, as long as, there's some correlation between how green the picture is and how much leaf area there is, perhaps GCC could be used to estimate plant water use. And I guess, Mustafa, you've done some work in like agricultural systems, or like the remote sensing of water use in agricultural systems. That's one of the methods.
Yeah.
It's to use some proxy of some index of greenness as a information on the development of the crop or something.
Yeah, there are actually some studies that like from different groups that they found a high agreement and high correlation uh, between the output from PhenoCam network, that is mainly GCC, and GPP of towers. So they are very kind of correlated to each other in different ecosystems. So basically, if there would be any kind of reduction in plant water use or any kind of water stress for plants, then there would be some reduction in GPP and also like transpiration of that plant, and then we are going to see a drop in GCC because the color of that plant is not going to be that green like it used to be before. So yeah, it can be used for water use purpose too. So
Yeah.
Oscar, yeah, go ahead.
Oh, well, I was just going to add on, Mustafa and I have just been talking about, deciduous forests and crops, but in evergreen canopies, like in evergreen conifer forests, you'd think that the greenness stays the same because they're evergreen trees, but actually the phenocams can detect slight changes in greenness, associated with changes in leaf pigments and sort of photosynthetic downregulation and upregulation. And so I think there's a um, nice paper from a year or two ago where they found that the onset of conifer transpiration that they estimated from dendrometer data, that actually correlated quite well with the increase in the conifer canopy greenness in the springtime. And so there's endless possibilities, I guess.
Yeah, for sure. So there's another question. Has anyone automated PhenoCam imagery to return crop height in agricultural settings?
Yeah, I mean, I guess if you could establish some calibration between, or if you're able to analyze the image to get how high the grass or whatever is, then calibrate that to measuring the height of the grass once every 2 weeks in the field, that would be possible. One method that a good amount of sites do is they'll have like a stake in the field of view of the camera. And I guess this is more of a manual approach, but they'll read off the stake. They'll use the stake to get the height of the grass from each image. And that's been done to, I guess, track the snowpack depth throughout the winter too, using these stakes in the image.
Yeah, and with some new tools like machine learning tools, there are some ways to automate that to get the like crop height and snow height as well. So it's a really nice field of study that so many. I'm not aware of any paper for using PhenoCam. I mean, I know for using like a snow depth, but not specifically for crop height. But I guess it can be used for crop height as well. Um, Another question, how about energy draw for those of us working with solar powered stations?
Yeah, well, I guess the good news is that I'd say the majority of PhenoCam sites run off of solar in the field. And yeah, there are a number of sites that are probably powered by relatively small solar power supplies. I don't have the specific power consumption off the top of my head, but I know at some of the more remote sites that my lab group manages, there are methods to minimizing the power draw, such as turning on and off the camera, before it's scheduled to take each image, or you can even limit the number of images the camera takes throughout the day. For example, most PhenoCams will take an image once every half hour, once every hour, but if you're really tight on power supply, you could limit that to just a couple images in the midday. Mustafa, anything to add on to that, or?
No, that was totally fine. So there's another, so let's go to the, because there are so many questions. Let's go to the next question that is saying not strictly PhenoCam related, but I am aware there are other methods to calculate transition dates other than the 10% threshold. For example, derivatives, rate of change. Would you know how they compare?
So, yeah, so if I were to provide a bit more detail on sort of the standard PhenoCam network algorithm, what we do is we fit a smoothing function, a smoothing spline to the annual cycle or the growth cycle, and then from the smooth GCC, we'll use this threshold method. The reason we've used this approach versus like fitting logistic functions and calculating finding when the curvature is this or whatever it is. The reason we've stuck to our approach is because we've found it's very flexible. This approach can be applied in not only, in deciduous canopies, evergreen canopies, grasses, even crops that might have multiple growth cycles throughout a year with, mowing and regrowth, for example. And so, we've stuck to our method because we believe it's the most flexible.
Yeah, and also PhenoCam network, we in all sites we provide GCC values too, and there are some studies that they use other methods than these other than these kind of threshold methods to get those like transition dates of start of growing season and end of growing season. But we also provide those thresholds of 10%, 25, and 50, just in case if someone wants to use those thresholds. So there is another questions. The seasonal GCC curve you showed for Bartlett has a steady summer decrease from the spring peak. However, I know the NDVI for that side is generally flat through the summer. How do you deal with that difference when comparing satellite NDVI and PhenoCam?
Yeah, that's a great question. That's a pretty good question. Yeah, and it comes down to the question of how similar GCC and NDVI are, and they are looking at different wavelengths, and so both are sort of greenness, but they're not identical. The GCC tends to be more indicative of the color of leaves, while the NDVI tends to be more indicative of the canopy structure, like the leaf area index, based on my experience and the work of others. And thus, in many cases, GCC is sufficient for evaluating satellite NDVI, but if you really look into it, that might not be always the case. And so that's sort of a caveat that needs to be considered. That being said, a lot of PhenoCams now are taking back-to-back, color and infrared images, like I showed on that one slide I had. And so one of the advantages of the camera-derived NDVI is that it might be more suitable for evaluating satellite remote sensing. Mustafa, do you think I missed anything in my
No,
response
that
to
was
that
great.
question there?
Just one thing to add. So, as you said, like GCC just relates to the color. So, like if you imagine a single leaf from like September to October in that site, when it turns from green to like orange, yellow, or red, its GCC value drops significantly, but NDVI, which uses the near infrared band, it's more a kind of photosynthesis rate, it's more related to like productivity rate and photosynthesis rate of that leaf, and that doesn't drop significantly compared to the color, and that's why NDVI doesn't change that much, but GCC drops significantly because color changes uh, from September to October in that site maybe. Another question, who is the author of the recent paper regarding evergreen forest dendrometer date correlated to PhenoCam data? You know, Oscar?
Yeah, thanks, Sharon, for your comment. I just typed it in the chat, so let me know if you got it.
Yeah. So another question, would it be possible to distinguish leaf color change being due to phenology versus due to stress drought?
Yeah, I think that's a great question, and I think that's something that I don't know, maybe hasn't really been looked at. What do you think, Mustafa?
Yeah, that's a very great question, actually. Yeah, I'm thinking about that. But I feel like maybe if you look at the seasonal cycle, that would be something more interesting. As we said about like that Bartlett site, like those color changes in like September, October, it's more kind of seasonal, yeah, phenological changes usually. But like if that would be like during the like growing season, like in July, August, or in like June. So that is more like water stress. If like other trees they are green, but a single tree is not that green, so maybe that can be an index of water stress for that individual tree, but actually not necessarily because that depends on other structural and functional traits of that tree too. I feel like color is one index that you can take a look, but there's always other things that you need to couple with GCC and kind of phenological changes to look at, I guess, water stress for individual trees.
Great. I think you guys have covered all the questions, a lot of questions. So, Mustafa, do you have your presentation ready as
Yeah,
well?
I feel like, yeah, are there so many other or we're done? Okay. Yeah.
I think
Yeah,
so.
yes, we are done. Cool. Yeah, yeah. We can start the next presentation then. Okay. Cool. Do you see my screen? Okay.
Yes.
So I want to give you a quick tutorial on navigating the PhenoCam website. Basically, the PhenoCam website is, the address is phenocam.nau.edu, and so this is where we store all of those images that Oscar was talking about, all of those 800 sites that we have in our network. So the first page is gonna be look like this. So sometimes we have an important update about if the server is down or whatever, if there is an issue, or if we have an important update, we put that on the first page. So now there is an update about like the new version of the cameras and the the guidelines to set up the Live 2 cameras, which is a new version of the PhenoCam cameras. So, and then there are two ways, like, let me see if sorry. And there are two ways to navigate through sites. So on the top of the first page, you see two tabs that are gallery and map. So you can see different sites in two ways actually here. If you click on gallery, you will see all sites that are available in the in the network. So these are all sites that we have in the in the network. So currently it is like above 700 sites, but I guess this is the previous version. It is now more than 800 sites on the PhenoCam network. And we have three types of sites in our network. Depends on how they follow our protocol and how the PIs of those sites collaborate with us. So we have type 1 that they follow our standardized protocol, and site personnel are actively engaged as PhenoCam collaborators. So it's basically we, uh, we, we trust those sites more than other sites because they follow our protocol and we know what they are doing. Type 2 are the type of sites that they are not using the standard camera, so maybe they are using other type of cameras that we don't suggest, but they follow our protocol and site personnel are involved. So these are type 2, so maybe a little bit more uncertainty, but still acceptable. Type 3, they have some deviation from standard protocol and no active collaboration with site personnel. We still support those cameras, but we don't have that much kind of confidence about those data because we don't know what kind of protocol they are following in their network. But most of our sites, I mean, probably more than half of our sites are type 1 that we can like trust them and trust those data. So in the gallery page, if you click on like each image or each site, you can go to that site, and then there are other ways to like filter those sites. So if you click on search sites in the gallery view, you can even filter them based on different things here. You can filter them based on site type, like if you want data that you're with high accuracy, you can just click on type 1 sites here. And then if you want just evergreen trees, you can just filter them based on evergreen trees. Or if you want like all grassland sites, you can filter them based on grassland sites. And even if you are looking for certain species, you can search for certain species here to like just get those species um, in that data. And also, if you want some sites that they definitely have flux tower data, you can filter them based on flux data as well. And all of these sites that they have EC kind of icon around them, it means that they have eddy covariance flux tower at those sites. And the second way is going through the map view. So if you click on map in the PhenoCam website in the first web page, then you can go through the map view. So these are some of the sites that we have in North America, and if you click on each point, you would see some summary of that sites, and then all of these sites are like in different colors: type 1, type 2, and type 3 that I mentioned earlier. So type 1 are green, type 2 are red, type 2 are yellow, and type 3 are red. So, and then if you click on one of them, then you can see that info. So this is one of my favorite kind of research sites in Arizona. So Walnut Gulch grassland site that I was working on that. And then you can even click to see different base maps. So if you want to have a different base maps here, and then also you can filter these sites here. Like if you are working on a project and you're working on a certain kind of criteria for your project, you can filter them here, like based on vegetation type, based on flux data, based on whatever that you want to filter, and then see how many sites are available. Like if you're writing a proposal or whatever that you are doing, you can filter them here and see how many sites are available for your purpose. And yeah, again, you can like click on any of them to go through that site and choose the base maps here. So if you click on any sites, then you would see these info for that site. So this is the Harvard site in in Harvard Forest, which is one of the long-term equivariance flux tower in the world. So, and also we have, it has one of the long-term record of PhenoCam data from 2008 to 2024. So on top left, you see some basic site information here, like site name, location, latitude, longitude, elevation, the number of images. So for these sites, there has been so far 200,000 images for that site from 2008 to 2024. And then you can also see more metadata of that site here. Then you can see the location of that site, and you can zoom in or zoom out. You can see all images that are available for that site for free here. You can see the latest image of that. So this is the latest image of that site that I took, I guess, a few days ago. So this is for October 9. And then you can go through different ROIs. So as Oscar mentioned, so we define some region of interest, that is ROI for each site, and that is usually based on what site PIs communicate with us, or based on what we think that can be useful for that site. So for example, if we click on one of these, uh, if we click on like browse images to see all the images of that site, we can see the all images of Harvard site from 2008 to 2024. Like in 2024, the images from January to October, like in January, there were more than 1,000 images in that site. And then if you click on one of those months, we can see all of the images of that month from the first day to the last day. So this is for October 2023 of Harvard Forest site, and you see the color changes. Like at the beginning of the month, they were all green, and then at the end of the month, they are turning to yellow and orange, which is really interesting. And then if you click on a certain day, like day 26 of October, so there are like 20 images in that day, and you can see all of the like sub-daily images of that day, that usually they are in half-hourly interval, but they can be different from site to site, from whatever the PIs of those sites want to have. Then if you click on one of those region of interest, you can see more info of like greenness of those ROIs here. Like if I click on like ROI page DB 1000, that is, so DB means deciduous broadleaf. EN means evergreen needleleaf. So we have a code for each vegetation type. So if we click on like DB 1000, we see the GCC of that region of interest. And and when I was talking about region of interest, so I'm talking about a polygon that we define to get those GCC values from that site. For example, for this site, we define this polygon in at the bottom of the page, because we wanted to avoid all of the things that are not part of vegetation, like this tools that is here. So we wanted to avoid that, and we wanted to avoid all of the background that are over there. We wanted to just focus on the foreground trees. So and basically this is something that PI of those sites ask us, and then we just define based on what PIs ask us. So this is the greenness change of that site from like 2008 to now. So and you see in some years, like here, like at the middle, like probably 2016 maybe, the GCC values peak is higher than other years. So probably that year was more productive than other years. And there are some years, like at the beginning when GCC was lower, the peak of GCC was lower. So this is basically the GCC time series of that side. You can even filter it based on specific years, like if you want to focus on specific years here. Here is the way that you can download those data. If you just download, if you click on provisional data, you can download all of these GCC data, and you can work on that data, plot it yourself, or use it for your purpose of that site. So, and this is the ROI mask of that site that we define. Like, for example, for this mask, this has been defined from the beginning of that site to now. But there are some sites that they have multiple masks. So depends on, so sometimes when there is a shift in camera or when there is a changes in like in purposes of that site, we define a new mask after a certain day to make sure that we are looking at the same thing. Like for this site, we have two masks. Like the first mask is from 2015 to 2016, and the second mask is from 2016 to now to like look at the same polygon to have high uh, confidence about the GCC values of that polygon here. Um, and then there's another way to look at those sites. So if you go to the PhenoCam Explorer, you can see different versions of PhenoCam data. The recent version is dataset v3. So and if you just select that, you can see all data that are available in that way too. And the cool thing about this tool is that like if you search for a certain site like Harvard here, you can see all of the transition dates of that site, like end of growing season, start of growing season, and all of the GCC values of that site, and you can download these transition dates here. So basically, these are all of the transition dates in different colors that you can see, and then you can even plot it in different ways or even covariate that uh, with, with, with satellite data like MODIS vegetation indexes, and then download all of these data together, and even you can compare like that questions that you asked about Bartlett, NDVI, and GCC. So it would be cool to like see them together to see what are the like specific dates that we see differences between those NDVI and GCC at that site. And then at the end, I just want to let you know that there are also other ways in the PhenoCam, in other tools in the PhenoCam website that you can download raw images and instructions to set up a new site that Oscar talked about that. Also, there are more info about PhenoCam website tutorial, the Python packages and R packages that are available for this data. So there are some R packages and Python packages that you can automatically download these data with those packages from PhenoCam network. And also there are some info uh, about K-12 lessons and plans and handouts and on the previous workshops and seminar about PhenoCam network. Yeah, I think that's it for me. Yeah, I'm happy to take any questions about how to navigate PhenoCam website.
Thank you, Mustafa. It was really helpful to understand how PhenoCam website works, really. And I see a couple of questions in the chat.
Mhm.
Um,
Let me see.
yes, I think the question is about the calibration of the PhenoCams, and if you have seen any drift of GCC over time.
Yeah, yeah, that's a good question. Yeah, there are so at in we have a few sites that we tried different kind of calibration methods for that site, like for red, green, and blue channel, and and then that was just for a as an experiment that we wanted to know how these values are kind of like how much uncertainty are in those values. But um, as far as I know, those experiments worked well, and they were high agreement between those kind of GCC values that they got from the camera with what they did over the kind of with their calibration method. But another thing is that like because GCC is a kind of it's the it's a green channel to the total of green, red, and blue. So usually if there would be a a systematic error or whatever, they offset each other, and usually GCC values is something that we can trust in a good way, but it doesn't mean that we calibrate all of our images or all sites, and that is something that sometimes there are some specific sites that PIs they do those calibrations themselves, but it is not in our protocol to calibrate all sites. So Oscar, do you have anything to add about that?
No, I don't. I don't have anything to add, but I did make a brief comment about it to Fred in the chat.
Cool, that's great. So there's another question. All of these site types are using star dot cameras if you're processing their imagery, correct? Yes. Oh, actually, not all of those sites, because there are, our type, there might be some sites in our type 3 sites that they are not using the star dot cameras that we propose. But I can say that most of our sites are using star dot cameras, and we process those imagery for each site. Yes. Um, there's another question. Only type 1 sites are using the standard camera. Type 2 sites. Oh, this is Oscar, sorry. So there is another question. How can we become a type 2 site? How are using a Raspberry? We are using a Raspberry Pi cam, but we are collecting at 30-minute interval and have maintained our camera. Sorry, I can't see. Our cameras with different amounts of usage. I think others have done more, but I can't think of anyone specific. I can't think of anyone specific right now. So, Oscar, I think you know some of those Raspberry Pi sites. Can you answer these questions?
Let me. This is in the chat.
Yeah. So so if they have a Raspberry Pi and they are collecting those images with Raspberry Pi, can they still use our network?
How to become. Oh, I see. How to become a Type 2 site. We are using a Raspberry Pi cam, but the camera's not networked. I think the simplest answer to your question would be to send us an email, send the PhenoCam team an email. So that's phenocam@nau.edu. And we might be able to archive your images. Although it usually is on a case by case basis. So we'll have to hear more about um, your site and the camera and the file names of the images and whatnot. So send us an email.
Nice. So there's another question about R packages for downloading PhenoCam data. I just replied with a link to that. Yeah, there are R packages that you can directly connect to the PhenoCam network and download data for specific sites or even specific vegetation types and filter them based on whatever you want. So I put the link in the chat. There's another question. Thank you for the presentation. How long will it take the images to appear in the PhenoCam website since submission of the details about the camera and the site? Oscar, do you have any?
How long will it take the images to appear in the PhenoCam website since submission of the details about the camera and the site? If I'm checking my email regularly, then probably within 24 hours. But yeah, within a week or so for sure. And if you need us to expedite anything, just send the PhenoCam team an email and we can do that for you.
Yeah, there's another question. On the question of drift, do you think that extracting a sky ROI is a good way to evaluate long-term drift? My thinking is that the sky condition will of course vary diurnally and seasonally, but should not drift over time. If a sky ROI shows directional change over many years, this could indicate drift in the camera sensor. This is very interesting, actually. It's a very probably a genius idea. Yeah, I mean, why not? I mean, that probably could be one way, but I guess I'm not sure if the clouds can have some effect on that, or So I feel like even sky shouldn't be something that always have exactly similar values. Yeah, this is something that can be like discussed or explored, but I guess, yeah, yeah, why not? That can be a way for sure. Do you have any idea, Oscar?
Yeah, there are actually some Japanese groups that have, I believe, done that way back in the day. Um, I don't, I don't, I don't know how successful they were or anything, but it has been done.
Mm-hmm. Cool. So I don't, did I miss any question or, okay, there's another one, I guess. My current sites are dry lands, so cloud are negligible. Okay, that's great. Yeah, I love dry lands. Yeah, probably you're in a good way, but I guess it depends what kind of dry lands. Like in Arizona, in the monsoon season, you get a lot of clouds. So, but yeah, but that's cool. Yeah.
Hey, any other question? Oh,
Yeah, I see something else.
yeah, sorry.
Just
Oscar
adding
is
to my,
new.
oh, this is Oscar, sorry.
Yeah. So yeah, any of you can just unmute yourself and speak up
Yeah,
if you want to
exactly.
talk to
Yeah,
speakers as well.
yeah, feel free to unmute.
Well, I don't have a question or anything, but so Mustafa, Darby, and I have given this talk, I think, 3 times already this year. Once at NAU here in Arizona, once at ESA in Long Beach, and once at the Great Thermal Bake Off, which was a FluxNet workshop. But anyway, today's webinar is definitely the sort of largest audience we've had and best engagement we have. And so I guess we just want to thank everyone for coming out and, Kung and the, what is it, the AMP team
AMP,
for
yeah.
AMP team for hosting us. And yeah, it's been great to share this resource that's out there that will surely help what everyone's working on.
Yeah, thank you so much everyone for joining us, and thank you Kung and Emerflux team to giving us this opportunity to like present for the community.
Yeah, I'd really like to thank you, Oscar and Mustafa, for today's really excellent sessions. And I myself learned a lot about PhenoCam and can't really wait to see what's the part 2 that is scheduled for the next Friday at the same time, 10 a.m. Pacific time. And today's session is recorded and will be uploaded to the Ameriflux YouTube channel in the coming days. So with that, I think, yeah, take care and see you next week, next
Yep.
Friday at 10 a.m. They're going to be hands-on session, PhenoCam hands-on session, right?
Yes, exactly.
All right.
Thank you so
Then,
much. Have a good
thank
day.
you so much. Have a good day, everyone. Bye.
Bye bye.